Matthew Perry, who starred as Chandler Bing on Friends, leaves a legacy that extends well beyond his acting. Throughout his life, he advocated for people battling addiction, drawing from his own lifelong battle with substance abuse. He spoke openly of his struggles, disclosing that at one point his intestines actually burst as a direct result of opioid use. In our lab, we're exploring something that is related to Perry's story of addiction, the connection between the gut and brain and the reason why the bacteria in your gut can be directly related to opioid addiction. Welcome to the Translational Addiction Podcast, where we decode the innovative research from the Lotfiepor lab at UC Irvine dedicated to translational addiction studies and developing effective treatments. I'm your host, Michaela Samorton. So I mentioned Matthew Perry in the beginning, and he struggled from a lifelong battle with substance abuse. On the show Friends, you can kind of even track this. He says when he was slightly overweight, he was drinking a lot of alcohol, and when he was underweight, he was taking a lot of opioids. He didn't start taking opioids until he was prescribed them after a jet ski accident, when he started taking Vicodin and liked how it felt, so he continued. At the most extreme, he was taking up to 55 pills per day. And one of the side effects of opioid use is constipation, which is the initial reason why he was hospitalized. He actually barely made it to the hospital when his intestines ruptured, and so they rushed him to emergency surgery, put him into a medically induced coma, and when he was released, the first thing that doctors prescribed him was opioids. And so this event brings up a couple of important concerns for both the American public and the healthcare industry. Opioid addiction is one of the most pressing public health crises in America today. And according to the CDC, fentanyl alone is the leading cause of death for Americans between 18 and 45 years old. And 1 of every 5 deaths in California is caused by fentanyl. So today, we will be discussing a review paper by Michelle Wren and Shardad Lotfiepor that analyzes how the interactions between the gut microbiome and the brain is affected by opioids and how this connection has the potential for therapeutic interventions for opioid addiction. And maybe in the future, we can even have the chance to stop addiction before it has a chance to start. So I mentioned the gut microbiome. It's kind of like an ecosystem of bacteria inside of your intestine, so including the large and small intestine, that's home to trillions of tiny organisms. It's also incredibly diverse. So imagine a rainforest of millions of animals and plants and insects, but this is a tiny rainforest inside of your stomach. And it's not only made of bacteria, but it also includes viruses and fungi and protozoa. And these organisms work together to help maintain bodily function. And so they help release certain hormones such as serotonin, they aid in digestion, and they also support immune function. And any disruption in these can be harmful to your health, including affecting circuits in your brain. So for example, consuming opioids causes interactions between the opioids and microbiome, which causes hormone imbalance. And the Wren Lotfiepor review will address opioid-induced changes to the gut microbiome through each stage of addiction. And so the gut microbiome actually connects to the brain through this powerful connection known as the gut-brain axis. And this connection is bidirectional, and so it's a pathway that's physically connected and chemically connected. So physically, the gut and brain are connected through a nerve called the vagus nerve, but chemically, they're connected through chemicals called neurotransmitters. And the brain can send signals through this pathway to the gut through these hormones or neurotransmitters, but the gut can also send signals back to the brain, affecting neural circuits. And so drug consumption leads to changes in the bacteria in your gut, which is referred to as gut dysbiosis, and this can lead to hormone imbalance. So for example, maybe decreases in dopamine and serotonin, which are important neurotransmitters that are involved in the reward systems in your brain. And any changes in dopamine or serotonin levels can be detrimental to health. It's even been associated with diseases such as Parkinson's. And so the goal of the Wren Lotfiepor review article is to establish a relationship between gut dysbiosis and the 3 stages of addiction. So the gut microbiome can be utilized as a therapeutic target in addiction treatments. So the 3 stages of addiction, as discussed in the Wren Lotfiepor review, are binge and intoxication, withdrawal and negative affect, and anticipation and craving. So we'll begin our review of the Ren Lotfipor article with binge and intoxication. The first major connection discussed in the Ren Lotfipor article is the gut microbiome's role in incentive salience, which is the mechanism by which addiction is developed through reward circuits in the brain. So here's an example of incentive salience. Imagine this. Every time you are in a certain room, maybe your bedroom, you take opioids. Eventually, your brain will form an association or connection between your room and the drug, specifically how you feel when you take this drug. And now every time you go to your room, you have the overwhelming urge to take the drug, and over time, you may even get a dopamine release from just being in your room because of the anticipation of getting the drug soon. This wanting feeling is incentive salience, and it persists even when the drug no longer produces the effects it initially produced. Incentive salience can be triggered by stimuli other than a place. It can be from people or drug paraphernalia, or even an internal trigger such as a certain mood. If you're interested in learning more about incentive salience, check out the Surgeon General report linked in the show notes on incentive salience. So reward circuits in your brain are controlling the stimulus-reward pathway on a neuronal level, and these circuits are generally studied relating to addiction as solely a neural issue, but in the Ren Lotfipor review, they attempt to extend this connection to the gut. So the article begins by reviewing obesity studies, which have established more extensive connections between the gut and brain, and the goal is of extending this connection between food addiction affecting the gut microbiome and neural circuits to opioid addiction affecting the gut microbiome and neural circuits. So foods that are high in fat and sugar activate the brain's reward system, which is not necessarily negative, but when you consume foods in excess, the microbiome in your gut changes, which in turn changes your brain function. And obesity studies have been connected to drug addiction because food addiction employs similar neural pathways. And the Ren Lotfipor article begins by reviewing other authors who have focused on these obesity studies and developed more established connections between food addiction and the gut microbiome, with the hope that we can eventually establish a connection between opioid addiction and the gut microbiome. So prolonged overconsumption of food is associated with decreased synaptic plasticity in your brain, which is the ability to create new connections in your brain, which is important for learning and memory. And the neurons in your brain need to be able to communicate with each other and establish new connections and strengthen existing connections for long-term and short-term memory to occur. And prolonged overconsumption of food has also been associated with increased vulnerability to anxiety-like behavior as well as disruptions in exploratory behavior in preclinical trials. So obesity studies are seeing a direct relationship between the gut and brain, and in the Ren Lotfipor article, they want to know how can this be applied to opioids. So opioids hijack the brain's reward system through incentive salience, which is how much you want something, for example, food or drugs, and the obesity studies have connected food addiction to changes in your gut microbiome, which affects neural circuits. And the Ren Lotfipor article is assessing, can this connection be applied to neural circuits involved in opioid addiction? So the next stage of addiction discussed in the Ren Lotfipor article is withdrawal and negative affect of opioid use. At this point, the user has built a tolerance to the drug, and the article is assessing whether gut-brain interactions are involved in the development of this tolerance. So tolerance to a drug in this context is analogous to this. Imagine the first time someone binges on alcohol. Maybe they have 5 drinks. This will have a significant effect on this person because they have no tolerance for alcohol. So they will feel extremely uncoordinated and their blood alcohol levels will spike. But over time, their liver starts to release enzymes that metabolize alcohol, and their body will develop the ability to metabolize more alcohol faster if they continue to binge like this. So as a result, tolerance to the drug is built because they can have more of it with diminishing returns. In the case of alcohol intoxication, someone who binges regularly may have the same 5 drinks that they did when they just started drinking, but now their blood alcohol level will be significantly lower because they've built a tolerance to this drug. And the Ren Lotfipor article wants to know how this kind of tolerance to opioids changes the gut microbiome. And if we can figure out the specific role of the gut-brain axis in opioid use and the factors that influence addiction, then the gut microbiome can be used as a potential therapeutic target and integrated with opioid regimens. So drug addiction is developed initially by drug use for a certain reward, but in later stages, it's defined as repeated use for distress avoidance. So this can even be relevant with a drug such as caffeine. Maybe somebody initially drinks coffee every day in the morning for more energy, but as they develop this metabolic tolerance, they need to drink more coffee more often to get to that same energy level. Later, as they continue to drink coffee every day, and at this point they've built a tolerance, instead of drinking it for more energy, they drink it to avoid getting a headache, which is the withdrawal symptom of caffeine. And this is because the brain and body have become so conditioned to this stimuli that they're constantly exposed to that the brain and body are now reliant on that stimulus. And so now, in the context of opioids, maybe this person takes opioids and they've built a tolerance and they have a decreased reward because they've gotten used to it and built that tolerance. And so they cannot reach the initial high that they felt the first few times they took the drug. And this inability to recreate that feeling increases stress and activates the stress response system in the body, which leads to increased stress and anxiety. And so if this person ever decides or has to stop taking opioids for whatever reason, and that stimulus is taken away, the result of that is withdrawal. Since the brain and body were accustomed to this constant exposure to opioids and now it's gone, these symptoms will have the exact opposite effect that opioid use did because the brain is trying to compensate for the sudden lack of opioids that it had grown accustomed to. So connecting this back to the gut microbiome, as discussed in the Ren Lotfipor paper, we know that microbiota help modulate stress responses as well as drug withdrawal-induced anxiety, which is why this review article is interested in using the gut microbiome as a potential therapeutic target. And we also know that imbalances in the gut activate stress response system, as we talked about earlier, and that is discussed in further detail in the review paper as well by Ren and Lotfipor. The stress response system can also be activated by imbalances in the gut, or it can cause imbalances in the gut. But either way, it's creating this feedback loop. So increased stress increases dysbiosis or alterations to the gut microbiome, which increases stress even more. So these stress-induced changes in microbiota can lead to GI disorders and mood disorders, and prolonged disruption of the gut microbiome has even been connected to depression and anxiety, as well as Parkinson's and schizophrenia. So the Ren Lotfipor paper assesses whether the gut microbiome may be utilized as a therapeutic target in the withdrawal and negative affect stages of opioid addiction. The final stage of addiction discussed in the Ren Lotfipor article is anticipation and craving. So in later stages of addiction, the gut microbiome plays a major role in relapse. And this can be through excessive incentive salience. So in the absence of drug, they really crave this drug, or when they're exposed to the drug or drug paraphernalia, they feel this uncontrollable desire to take that drug. And if they do get that drug, they have the diminished reward system function. So it's not the same effect that they used to feel, and they always want more and more of it. And this cycle increases stress, leading to increased drug seeking. And the 3 stages of addiction may continue in a cycle at any point in this individual's life if they do relapse. And so the Ren Lotfipor article reviews whether the gut microbiome can be used to stop this cycle as a therapeutic intervention. And so They do outline a few experimental techniques used to study the effects of opioids on the gut microbiome, and we will not cover all of those techniques today in this podcast, but we will outline one. So, to study the role of the gut microbiome in opioid addiction, mice are used as a model organism, and these mice have been bred or genetically altered to contain a certain type of gut microbiome. And in order to study microbiome changes in the microbiome of these mice, researchers can use fecal microbiota transplant, or FMT, which means that they will actually transfer the fecal microbiome of a mouse infected with a certain drug or disease or bacteria to another otherwise healthy mouse. And the results will tell them what the effect of that intervention was on the microbiome of the healthy mouse. And so FMT is actually not a novel intervention. It has been used as a therapeutic intervention for C. diff patients. And so C. diff is a bacteria that can be contracted through physical contact with contaminated surfaces, and it is an infection of the gut that leads to diarrhea, fever, and abdominal pain, as well as other GI issues, including colon inflammation. And one of the treatments for patients with C. diff is to use fecal microbiome transplant. So we take the stool of a healthy person and transplant it into the person with C. diff. And this will allow the bacterial species to colonize and repopulate the intestinal flora with more diversity. And that in turn allows the patient to fight off C. diff and regain the ability to basically poop effectively. And this intervention may seem slightly off-putting, but it has a 90% efficacy rate. And sometimes patients can even be prescribed pills with fecal matter in them, and they'll release the fecal matter into the intestinal tract and allow the flora to become more diverse. And so FMT has also been considered as an intervention for depression and anxiety, as well as other psychological disorders. But the focus of the Wren-Lotfeport paper is if FMT can be used as an intervention for opioid addiction. And so individuals who regularly take opioids have reduced diversity in their gut microbiome, and recolonizing their microbiome with more diverse species has the potential to reduce opioid-induced constipation as well as reduce anxiety and depression. And so one of the studies outlined in the Wren-Lotfeport paper takes the fecal microbiome from a mouse with tolerance to painkillers and transfers it to a healthy mouse, such as we described earlier, and they found that tolerance is transferable via the fecal microbiome. And so if you have a gut microbiome of a certain composition, that can give rise to drug tolerance. And this connection supports the idea that the gut microbiome plays a significant role in drug addiction, and that restoration and increased diversity within the gut microbiome can potentially aid in opioid withdrawal. We just discussed the role of the gut microbiome in the 3 stages of opioid addiction. So binge and intoxication, where we explored the role of incentive salience in developing addiction; withdrawal and negative affect, which is what happens to a person when they stop taking a drug that they've grown a metabolic tolerance to; and anticipation and craving, which can manifest as excessive incentive salience or the overwhelming urge to take a drug. So now we will briefly outline the mechanisms by which the gut microbiome affects opioid use behavior, beginning with neuroinflammation. So when there's any disruption in the communication system between the gut and brain, either through the vagus nerve or neurotransmitter disruption, the immune system response is triggered, and this can cause inflammation in the brain as well as disruption in the gut microbiome. And this inflammation can also be exacerbated through a Western-style diet or certain meds or stress or autoimmune disorders that deteriorate the intestinal barrier. But it can also be caused by opioids. And this neuroinflammation can lead to dysfunction in neurons that release dopamine, which will end up dysregulating the reward systems and result in chronic pain, depression, and sometimes even opioid addiction if a person takes opioids to relieve this pain or depression. And so Ultimately, the negative effects of opioids may even be increased by neuroinflammation in later stages of addiction. The second mechanism we will discuss is the role of microbial metabolites. And so your gut breaks down nutrients for your body to use through a process called metabolism, which is the breaking down of energy from food into components that your body can use to function. And this process is assisted by the bacteria in your gut microbiome. One of the products of metabolism is called short-chain fatty acids, and these are associated with neural function. For example, they can influence learning and memory as well as alleviate stress. And it is important to note that fecal microbiome transplant is not the only intervention that we can use for the gut microbiome. The Ren-Lotfipor paper also reviews research on the effects of short-chain fatty acids. And so these have been known to restore certain cells that help maintain homeostasis in the brain, and these cells are called microglia. And short-chain fatty acids help restore any abnormalities in these microglia in mice, and they've also been seen to reverse enhanced reward sensitivity to cocaine, which has been seen in gut bacteria depleted mice. The third mechanism we will discuss today is the role of serotonin. And so microbiota regulate the synthesis of a precursor to serotonin, which is called 5-hydroxytryptamine, or 5-HT for short. And this is significant because any dysfunction in 5-HT can lead to GI and mood disorders. 5-HT is also directly involved in pain perception and regulation of gut secretion and motility, but the main signaling function it is involved in is regulating mood and cognition. And the relationship between gut bacteria, serotonin synthesis and signaling, and mood is crucial to note to consider in addiction and addiction-motivated behavior studies. The final mechanism we will discuss today is the role of microbial diversity and presence of specific species. And so any imbalance in the bacteria in your gut can disrupt immunity and nutrient absorption. And any overgrowth of bacteria can lead to secondary infections, including C. diff, as we discussed earlier in the context of fecal microbiota transplant. It is beneficial for there to be a high diversity of microbiota in the gut, so you want many different species to be present, and this will help to decrease anxiety and stress-induced hormone release with increased diversity. Throughout the Ren-Lotfipor review, there is an apparent relationship between gut bacteria and tolerance to opioids. So in the future, researchers would need to evaluate how the gut microbiome modulates the opioid reward and reinforcement systems, as well as evaluate how gut bacteria changes can affect the circuits in the brain involved in reward processing. And studies would also need to be conducted on how dysbiosis is involved in the feedback loop that exacerbates stress, leading to anxiety and depression. And these are all open areas of research today that have the goal of opening up new pathways for treatments. Because if we can understand how these networks work, then we can take steps towards problem solving. If you're interested in learning more about the connection between your gut and opioids, check out the infographics and papers on the Lotfipor Lab website, linked in the show notes, or the National Institute on Drug Abuse to learn more about how addiction is treated and how science is evolving to fight against this crisis. The Michelle Wren and Chardad Lotfipor paper discussed today is called The Role of the Gut Microbiome in Opioid Use and can be found on the Lotfipor Lab website. These resources dive deeper into the lab techniques used to establish biological connections and explain further how opioids lead to neuroinflammation and alter microbiota. If you or someone you know is struggling with addiction, you're not alone. Please contact your healthcare provider or refer to the resources linked in the show notes. The Translational Addiction Podcast is a production of the Lotfipor Lab at UC Irvine, exploring how to understand addiction and develop effective treatments. See you next time, and thank you for listening.